she and I have a very, very strong bond that goes back before that. And that is our...
I'm sorry to interrupt. I want to remind Farzan to record this, please.
I am recording. Thank you.
You don't trust him, do you? So when Al Silverson, who was both of our thesis advisor, was asked early on in my graduate career whether he would take me as a graduate student, he told Brian Maloney, or he told somebody, no, I think he told Tenny Maynard, Don Maynard's wife, that he would never take a woman in his lab as a graduate student because she would never get a job. Now, he apparently, you know, he had probably one of the most successful set of female graduate students of any person of his generation, because I was there at the same time Cheryl Glazer, who was a physics graduate student who was doing connectome work way before anyone knew what that was. And then there was me, and then there was Brenda Claiborne was before Gina, right? Mm-hmm. Brenda became very successful as well. She moved into the hippocampus, and then Gina, and who else? There was somebody else, I don't remember. But certainly the experience of going through Al's lab was one that we've shared, and I think that has shaped both of our sort of independence. Either it required somebody who was very independent to survive there, or that experience shaped our ability to be independent. That's probably both. So when Gina started in my lab, I was extremely happy because she was a very exciting, wonderful person. And she did some of the most beautiful work as a postdoc of anybody I've had go through my lab. And I should say that was one of the golden eras of the lab. And then she went on to her own lab, where she has actually become an extraordinary star in her own right. Now, I'd like to say that one of the things that characterized Gina as from very, very early on is that she is one of the people who has the clearest ability, or the ability to see a clear path in search of the answer to a problem. And she's quite remarkable in that way. Her work has always been characterized by her ability to go in a straight direction and see the essentials. So she did some very lovely work at the very beginning of our studies of homeostatic regulation of intrinsic excitability. And within a year after starting her own lab, she and Sasha Nelson had really started, or a year and a half, and started to develop the concept of synaptic scaling, which sort of built after the transition to synaptic transmission and mouse and rat. Now, many people would be very hesitant to switch from the STG to mouse and rat, or rodents, it was rat at the time, without any formal training. She just did it, you know, and I think she's one of the embodiments of someone, and I just like to say she was one of the people who was able to just figure out how to do something, whatever the something is. And it's, I think, one of the reasons why she's been so successful. She's completely unfazed by what she doesn't know. And she has constantly kept moving as the field gets moving, and start pushing the field as the field gets pushed. So it gives me great pleasure to welcome you back to your old family, your old scientific family. And we're all extremely pleased to have you with us. So it's all yours. Share your screen.
Great. Well, thanks. Thanks to all of you for inviting me. It's really an incredible pleasure to be back and see all of your faces and great friends from, you know, long ago. And I was actually, as Eve was telling stories about Al's lab, I was thinking about Mikey and how he really helped me in my first rather difficult moments in Al's lab. So anyway, you guys have all been like an amazing group of people to know over the years. And I just want to say that the mammalian synaptic plasticity field is not as nice. People are just not as nice. But anyway, okay, so I'm going to share my screen. I wasn't, completely sure what I was going to do.
Actually, doesn't matter.
But I'm just going to give you a kind of, overview of the journey that we've taken as we've tried to follow this path of understanding homeostatic plasticity and what it actually does in cortical circuits. This concept of homeostasis, of course, is sort of deep and intrinsic to our understanding of physiological systems. When I joined Eve's lab, this was not on my mind at all. My plan had been to study neuromodulation, and I was pulling neurons out of the animal and putting them into culture with the idea that, we would isolate them and be able to more easily study modulation of currents and things like that. And weird stuff started happening. And the properties of the neurons were changing in interesting ways. And at the same time, my desk was next to Gwen Lemassance, and he was a good friend of mine, a postdoc in the lab, and he was working on these beautiful little models of activity-dependent regulation of bursting patterns. And I was looking at my data and looking at Gwen's model, and the lights kind of went off, and so I guess the beauty of this whole sort of concept of homeostasis has really stayed with me. I think I really had no idea how rich this was going to be and how long the journey would be, but it's been a lot of fun. So I don't have any photos of Eve from back when, but I do just want to say she's been an incredible mentor over the years, and despite the awkwardness of having one of her postdocs stay on, she never gave any hint of finding that irksome at all. She's always been an absolutely amazing mentor and really helped build this incredible community at Brandeis in neuroscience, which has been amazing. I really like this picture from just a couple of years ago of a group of now senior women. We all sort of started pretty close to each other, and Eve really mentored all of us. And one thing is kind of interesting is that I've actually published papers with all of them. So with Eve, of course, but also with Piali, who works primarily on C. elegans, with Leslie, who works primarily on Drosophila, with Susan, who also works on mice. So it's really been just a wonderful collaborative place to do science. And that's part of what made it actually pretty easy to switch from working on, spiny lobsters to working on rodents in my own lab. I didn't know what I was doing. Originally I thought I would keep working on stomatogastric neurons, but it turned out that with some help from Susan, we could very quickly get rat cortical neurons, like hundreds of thousands of rat cortical neurons in culture. The sort of collaborative environment at Brandeis and just the willingness of people to help me figure stuff out really made that transition possible. Of course, the other major influence and collaborator that really made all of this work was Sasha. And this is not exactly when we started, but around that time, very long ago, black and white. But when we started at Brandeis, I don't know if all of you know this, but we actually started sharing a tenure track position. So we had the wonderful situation of not having job offers on the same coast. Brandeis sort of saw an opportunity. And so actually Sasha proposed to them that they offer each of us half a tenure track position. And so that's how we got started. We each had half a job. We had half of a $150,000 startup, which meant we each had exactly enough money to build one electrophysiology rig. Half of a not very large lab space. But that was, what it took to get going. And so Sasha really and I started out working very closely together, and we collaborated. I want to say, yeah, we published something like 27 papers together. We've also collaborated on these two kids who at this point are actually young adults. They think they're adults. They're sort of adults. Anyway, so Sasha was a really important collaborator, especially back at the beginning, but we've continued to collaborate on a lot of projects. Okay, so so from there, you know, with one electrophysiology rig, I sort of set off into this odyssey of trying to understand homeostatic plasticity in cortical circuits. And the motivation for doing this was really, I just had this intuition that these beautiful principles that applied to the regulation of intrinsic excitability might also apply to the regulation of synapses, because of course the excitability of a neuron depends not just on its intrinsic conductances, but also on the balance of all of these excitatory and inhibitory synapses that it's getting from all of these other members of these complex circuits. Oh, and by the way, I just meant to say that, you know, I'm just going to ramble. I'm going to talk about a couple of big problems that we've worked on. We'll see how far I get, but just interrupt me and ask questions. I mean, I know this is a different, you know, it's, even though a lot of the ideas are going to be very familiar, you know, it's a different system. And so anything, I'm really happy to just go sideways and talk about anything that you want to. So if you want to ask questions, just unmute yourself and ask. Okay, so there's sort of two big problems that have really motivated the work we've done over the last 25 years or so. And one of them is just cartoon in the sort of thought experiment, if you will. And it's really the question of how it is that you could set up a neural circuit so that it can maintain stable function and can faithfully propagate signals, which, you know, is just the simplest thing the circuit ever has to do, let alone process those signals or do more interesting computations. And, you know, the thought experiment is pretty simplistic. It's just that if the gain of transmission from one layer to the next is a little greater than 1, then your signal is going to keep growing as you traverse the layers. And pretty quickly, you end up in a situation where every neuron is just firing all the time. And you've built a hyperactive circuit. And if you do the opposite, you know, if you make sure the gain is less than 1, then you've got the opposite problem. You've basically built a kind of catatonic circuit. And in both of these states, information, you know, is either lost here or it's just lost because you can't faithfully propagate it. And so what this thought experiment suggests is that to allow circuits to, you know, just do the simplest thing they ever have to do, faithfully transmit information, you need to make sure that the gain of transmission within these networks is tuned pretty close to 1. And the other really profound problem, of course, is that you can't just set all of these synapses to the right strengths and then let the thing go, you know, you're done, because experience is constantly changing the strength of those connections as a function of learning and experience-dependent development and all of that. And so what that really suggests is that these networks need mechanisms that actually dynamically tune the strengths of connectivity or excitability to keep it within some kind of functional range. Okay, so this is a pretty familiar concept to all of you, I think. So this is one idea that was really sort of driving our thinking. But there was another idea, and this was actually sort of introduced to me by Larry Abbott, who was my, you know, one of my co-mentors when I was a postdoc at Brandeis. And he sort of suggested I go read a bunch of papers on, you know, Hebbian plasticity and some of the pitfalls of using those mechanisms in computational models. And it turns out that these sort of Hebbian plasticity rules, like long-term potentiation, are positive feedback rules that themselves introduce really major problems when you put them into computational models if you don't build in some sort of countervailing homeostatic force. And basically it's because what these rules tell you to do is, if there's a correlation between pre and postsynaptic firing, you should strengthen that synapse. But then when you strengthen the synapse, you increase the correlation. And so the synapse will just keep strengthening until it reaches, some bound. And the problem is actually worse than that. And this idea is sort of cartooned here because it leads to loss of specificity. So if you potentiate one synapse in particular, you end up driving the postsynaptic neuron more strongly, and that makes it easier for other neurons to make it fire. And so the correlations here will start to go up, even if they're not signaling anything about the environment, correlations in the environment. And so your potentiation can spread, and essentially you can end up with every synapse in your network locked up at some maximum value. And so what Larry was pointing out to me was that computational modelers would build in some sort of normalization rule, some sort of synaptic normalization rule that would prevent this unconstrained potentiation from happening. And so one of the ideas we sort of started off with way back at the beginning and haven't actually been able to test until very recently was this idea that homeostatic regulation of synapses, homeostatic synaptic normalization, if you will, might act to constrain long-term potentiation and thus have an influence on memory formation. So if we get to it today, I'll tell you about some recent work we've been doing that comes back to this idea. Okay, but first, this more sort of general way of framing the problem. One way that you could solve this problem is if individual neurons in your network have a way of sensing how active they've been and adjusting their excitability to keep their activity relatively constant. So the idea was really that neurons might regulate their excitability around some kind of activity set point. So when we first started working on this idea, we used these dissociated cultures from visual cortical neurons. So we took them from rat visual cortex, just postnatal animals, and we plated them onto beds of astrocytes, and they would form these nice networks that become spontaneously active. And so this was nice because we could pharmacologically manipulate their activity and then ask whether this produces any sort of compensatory changes in synaptic strengths. So we could pharmacologically raise firing. And one thing that we found is that if you did this, for instance, you could block a fraction of inhibition in these cultures, do that chronically. But even though inhibition was still blocked, firing would actually come back very close to where it was before. And other people like Juan Barón did an experiment where he genetically reduced activity in a subset of neurons and again found that those neurons would start to fire again, even though that manipulation was still present. And more recent work from Inosletsky's lab has shown this in hippocampal networks. So a whole lot of work from many labs now actually showing this, demonstrating the same sort of idea that in these dissociated networks of neurons, they really behave as though the network has an activity set point. And so one possibility is that this is really determined at the level of individual neurons, that individual neurons have a set point. When you perturb them, they come back to. And that's an idea that I'll sort of draw out for you in this talk. So this is sort of the idea in cartoon form. There's some target activity, some target firing rate, let's say. It doesn't have to be firing rate, but in cortical neurons, which are a little boring and tend to fire pretty tonically, firing rate is sort of a good proxy for overall activity. So the idea is, if they fire more, synapses get potentiated, push them out of that target firing rate, they would somehow reduce excitability to bring them back, and vice versa. So it turns out, just summarizing like a huge amount of work from, again, from our lab and many labs, that there's a whole family of homeostatic plasticity mechanisms that contribute to this regulation of network activity. So if you do a sensory deprivation experiment or you throw tetrodotoxin onto these cultures to block spikes, what you see is a whole coordinated set of compensatory changes. So you see changes in synapses, both excitatory and inhibitory, and you can see changes in intrinsic excitability, and these are all in the right direction to contribute to that restoration of activity. And you know, there's a number of reviews in different systems on these sorts of problems that, again, it's a very general phenomenon, but the exact mechanisms, the exact compensatory mechanisms that get used probably depend critically on the nature of the circuit, what it is that the circuit is actually trying to conserve, and you know, cell types and so on. So there's a lot of complexity here. And we've sort of come to think of these as being modules, if you will, that can be sort of mixed and matched and expressed in different cell types to sort of achieve whatever the homeostatic goal of the network is. But I'm going to talk a fair amount today about one form of homeostatic plasticity, and that's synaptic scaling. And this is one that we've studied a lot and the field has studied a lot, and we probably have the most insight into in terms of overall mechanism and function. And what we showed a long time ago is that if we, for instance, bathed these dissociated cultures in TTX to block activity and then came in and measured the strength of excitatory currents, we found that all of those excitatory currents, well, the distribution of amplitudes of those currents was increased. Basically, it was scaled up. This is actually one of the most fun experiments I did in my own lab, I think, was putting TTX on these cultures with this idea that we might see this, and then going back and recording from them and patching onto these neurons and finding just these enormous synaptic currents. And that was a pretty exciting moment when it seemed like there was likely to be something there. Okay, so basically this is a bidirectional process. If you increase activity, excitatory synapses get scaled down in amplitude and vice versa. And it's got a number of properties that are pretty interesting, and they differentiate it from you know, more classic forms of synaptic plasticity like long-term potentiation. So first of all, they're really slow. It takes many, many hours or days to induce these changes. So they're slow and they're cumulative. You know, the longer you deprive the cultures, for instance, of activity, the stronger synapses get. To a sort of first approximation, they seem to operate globally. So rather than being synapse-specific changes that affect just a few synapses onto a neuron, they seem to affect most of the neurons, most of the synapses onto the neuron. There's some interesting complexities once people started studying this in vivo. That you know, like all of these things, we have to well, whatever, the in vivo situation layers on a bunch of very interesting complexities. I'll talk about some of those a little later on. But one of the nice things about, on a theoretical level, about this idea of scaling the synaptic weight distribution up or down is that this means the relative strengths of synapses are not affected by this process. And so if you were to store information as differences in the weights of those synapses, as you know, is one of the theories about how learning works, this mechanism would allow neurons to regulate their overall excitation without affecting those synapse-specific differences. Okay, so we know, again, based on the work from lots and lots of labs now, we know something about the signaling pathways, but not all that much. You know, it turns out to be really complicated, and actually these changes in synaptic weight probably involve, you know, every single cell biological process in the neuron. So they're calcium dependent, transcription dependent, and the net result is that they alter, these changes in activity alter the trafficking of glutamate receptors to synapses to modulate synaptic strength. But one thing that I want to emphasize is Again, to a first approximation, this process seems to really be cell autonomous. So for instance, we can block spikes in an individual neuron. The rest of the network is still firing away and doing its thing, and we can induce synaptic scaling in that neuron. So this sort of classic form of synaptic scaling seems to really, the neuron seems to be paying attention to its own activity. And this sort of becomes interesting later when we think about how this plays out in vivo. Okay, so many years ago now, we decided we really had to go beyond this culture system, which was great for working, for showing these sort of the existence of these fundamental processes and for working out mechanisms, but isn't so great if you're interested in how these play out in a real brain attached to a real animal that's actually out and behaving in the world. So we thought we'd co-opt this very classic visual system paradigm for manipulating visual experience, going all the way back, of course, to Hubel and Wiesel, and that is just to lower a visual drive to one hemisphere of visual cortex. As a kind of, initially we thought a kind of analog to, throwing TTX onto a culture. And one thing I should tell you about rats, because I don't know how many of you have stared at rats as much as I have, but their eyes are much more on the sides of their heads than a primate's. And the eyes mostly see different parts of the visual world. So input from this eye is mostly mapped onto the contralateral hemisphere into primary visual cortex and vice versa. So there's a big region of visual cortex that is only driven by the contralateral eye. There's a small portion, because the animal is about 30 degrees of their visual field overlaps, so that gets mapped onto binocular cortex. But this is nice for us because we can manipulate one hemisphere, leave the other unaffected, and we have this very nice sort of within animal control for a lot of our experiments. Okay, so we've done this experiment a number of different ways. You can block the optic activity in the optic nerve, or you can close an eye via lid suture, and they all show something very similar. Other people like Tara Keck have used focal lesions of the retina. And again, you see something quite similar, which is that in the deprived hemisphere, in this case, we're recording from these layer 4 star pyramidal neurons. So this is most of the thalamic inputs to cortex land here in layer 4. So it's sort of the primary input layer. And if you record from these guys, what you see is that the synaptic currents in the deprived hemisphere are bigger than in the control hemisphere. And you can remove that visual deprivation, and this reverses. And it has the same sort of scaling property that I was mentioning before. What this shows you is the distribution of these, the amplitude of these quantal events. These are miniature excitatory postsynaptic currents, which is just a sort of classic now way of studying synaptic scaling. It allows you to study, to measure the postsynaptic strength of many, many synapses onto a neuron. So it gives you kind of an overview of synaptic strength onto that neuron. And what you see is that in the deprived hemisphere, this distribution shifts towards larger values, and you can scale that distribution down multiplicatively and recover the control distribution. Okay, so synaptic scaling is present in V1, as are other forms of homeostatic plasticity, it turns out. But does this mean that neurons in vivo actually have a firing rate set point? So this is something we had really wanted to ask for a long time, but it's a difficult experiment to do. But a number of years ago, I was lucky enough to have Keith Hengen join the lab. And he was, maybe as Eve said of me, very fearless. And so he just decided to tackle this problem head on. And we thought about it a lot. What's the best way to measure activity, calcium imaging, et cetera, really just wasn't going to work because if we were going to ask a question about firing rate set points, we really needed to measure firing rates. Calcium was not a good enough proxy. Okay. So what this meant was we basically had to figure out how to record from the visual cortex of freely behaving animals. And this wasn't something my lab had ever done before, but Keith just sort of jumped in with both feet. We also, again, had amazing collaborators at Brandeis, in particular, Steve Van Hooser, who's just instrumental in helping us get the analytic pipelines up and running, and also Don Katz, who really helped us with these chronic recordings. Okay, so what Keith did is this is an early version of the experiment, but it just sort of gets across the idea. We're doing all of these experiments during the classic visual system critical period. So these animals are pretty young, they're juveniles, so they're not that big yet. And what Keith did was to implant multi-electrode arrays into both hemispheres of monocular visual cortex, and then wait for the animals to recover, and then put them into this arena, plug them in. The experiment now is they have a commutator so they can run around and it doesn't tie up the electrodes. And they have a sibling for interactions, food and water, and they get a new toy every day. So it's kind of a enriched environment. And they're just running around doing their thing. And we're just recording the activity of all of the neurons that we can sample across the layers, although we have information about what layer we're recording from. So this shows you an example of one of these recordings. That's a heat map of the firing rates. And this is 200 minutes of recording here. And one thing you can see, I think, right away is that each neuron in this ensemble has really different mean firing rates. This is actually something that's been observed pretty much time anyone puts electrodes into any region of cortex, even if you're recording from the same, let me back up for a second. Technically, let me tell you that most of the data I'm going to show you today all comes from neurons with a certain spike waveform, which are regular spiking neurons. So we have excluded from this analysis fast spiking GABAergic interneurons, which have a very distinctive waveform that you can detect extracellularly. So most of the cells I'm telling you about today are these excitatory pyramidal neurons, but there's some regular spiking GABAergic neurons mixed in at low frequency that we can't detect. And we know, again, what layer we're recording from. So even if you're recording from the same layer, it turns out neurons sitting right next to each other, say layer 2 pyramidal neurons, can be firing at very, very different mean rates. And their firing fluctuates, but it fluctuates seemingly around that sort of mean rate. Okay. So now what's the experiment? The experiment is to record a couple of hours of activity a day. So put the animal in the arena, plug them in, record for 8 hours, same circadian cycle each day. Do this for 3 days to get a sort of baseline level of activity. And then what we're going to do is close one of the eyes. And we're recording from both hemispheres, so we now have a manipulated and a control hemisphere. And we're just going to wait. And what we thought would happen is we'd close an eye and we'd withdraw visual drive and firing would drop. And then maybe over time, if we waited long enough, and this idea of firing rate homeostasis was right, maybe activity would come back. So this is what we saw. Now, in this experiment, we're not trying to follow the same neurons over time, but I'll get to that in a minute. So here, we're just recording, whatever ensemble of neurons we can record across these different days. And what we saw is that this is baseline in green here. On the second day of binocular deprivation, this whole distribution of mean firing rates shifted to the left towards lower values. So as we predicted, firing rates drop. But if we came back and recorded a couple of days later, what we saw is that that distribution actually came back to become statistically indistinguishable from the control distribution. So that told us that activity at the level of the network was being restored remember, the eye is still closed at this point. And it also told us that the distribution of firing rates, which again follows this, it's very broad distribution. It's actually a log normal distribution. That distribution is also being restored. And so that suggested one of two things could be going on. One is that there was some mechanism at the level of the network that really constrained that distribution. And that could be true, still could be true. But we don't have a good candidate for such a mechanism. But the other maybe simpler possibility is that individual neurons might be, their firing rates might be dropping, but then perhaps coming back very close to where they were before. So in other words, that neurons are really behaving as though they have an individual firing rate set point. So that's the idea that Keith wanted to test next. And to do this, what he had to do was actually follow the same neurons over time. And this is a long experiment. This is a 9-day experiment here, 12 days later. So actually at the time we started this experiment, this really hadn't been done before. So Keith spent a lot of time trying to sort of optimize these recordings and was able to get them stable enough that he could in fact do this. So this is to convince you that we can follow the same neurons over time. This shows you two, this is the principal component analysis of the spike waveforms from two neurons that we were recording from the same electrode. And the color coding shows you the spikes by day. And it's just to show you they're all intermixed in these two separable clouds. So the clouds stay pretty well separated over time. And this shows you the centroids of each of those clouds, which also stays pretty stable. And this also shows you the spike waveforms, which you can peak scale. There's some small changes in amplitude without much change in the waveform, and overlay them, and you can see they remain distinct over time. So
Gina, I have a question.
Yeah.
So you have this activity that you record in visual cortex, but at the same time, one of the few things I know about mice is that they are nocturnal. And so I'm wondering, as you're recording for these prolonged periods of time, what are the lighting conditions that the mice experience?
Okay, that's a great question. I'm about to show you that. But first of all, I will just tell you that rats are not nocturnal. They sleep more during the day. They spend about 60% of their time during the day sleeping and about 40% of their time during the dark phase sleeping. So there's a difference, but it's actually not that extreme. And they're very, they do not have consolidated sleep, which is something I'll talk about depending on how our time goes. But it's a great question. How does this activity change across light and dark cycles, circadian cycles, sleep and wake cycles? And that's something we were really excited to look at with these data sets, because one of the beautiful things about being able to follow neurons over time is that we have all of this incredible data about the behavior of the animals that we can then sort of correlate with these activity patterns. So we can follow neurons over time, and not a lot. So this is a pretty low yield experiment, but it's a doable experiment. And this starts to get at Astrid's question. So what this shows you is the light dark phases over time of a group, an ensemble of neurons that we were recording from visual cortex. And I think there's a couple of really striking things about this. So this is just control conditions. We haven't done anything. The animal's just running around. And what you can see, again, this is a log scale. So you see this really wide distribution of mean firing rates. The other thing I think you can appreciate is that the firing rates are actually, the mean rates are not very different between light and dark. And actually this kind of blew our minds when we first saw this. We spent a lot of time digging into this, and we have a whole separate paper on this in PNAS if you're interested. But the bottom line is that mean rates are remarkably insensitive to light and dark, even though this is primary visual cortex. They're remarkably insensitive to whether the animal is asleep or awake. So there's, 5% differences in mean rates across, non-REM versus REM. It's really very, very tiny compared to this, many orders of magnitude difference in the mean rates that neurons have, even if, they're two layer 2-3 pyramidal neurons sitting right next to each other. So this, I think, is a really kind of remarkable feature of visual cortex. And one might ask, is this unique to visual cortex? Actually, Ben Solvecki's done some very similar experiments in motor cortex and seen something really similar. Not the light and dark, but the fact that individual neurons seem to have these really stable firing rates over time. So we think, the rapid fluctuations, which you can't see here because we've smoothed them out, are signaling something about the environment, about the visual environment, but the mean rates really are insensitive to whether the animal is seeing or not seeing. Okay, so that was kind of surprise number one. So now, the next, the other thing I just want to say is just, the sort of order of neurons and the distribution stays really stable over time. So this really kind of suggests that even in these juvenile animals, the activity has already kind of reached some kind of stable state where individual neurons are really able to maintain a very stable mean rate. All right, so what happens now when we do our monocular deprivation experiment? And this shows you two neurons that we're following over time. This is when we start the monocular deprivation. And one thing I want to point out is that when you close an eye, you do not see an immediate drop in firing. And this maybe doesn't surprise you now that you know that the light dark, the activity across light and dark is really not very different. So it's not the closing of the eye and the withdrawal of sensory input that drives this drop in firing. It's actually an active suppression of activity. And it seems to happen due to Hebbian LTD-like mechanisms and changes in inhibitory circuitry within V1 that sort of actively drive this suppression. And then what you see is this recovery of activity. So in both of these neurons, activity starts to come back. And in these two examples, it looks like it comes back pretty close to where it started, even though, again, log scale, so they have really, really different mean rates. Okay, two examples, but what about the whole population of neurons? So this is one way of looking at the data, where what we did was we plotted the mean firing rate of the neuron at baseline against the mean firing rate during this early monocular deprivation when firing rates have dropped. And in the control hemisphere, what you see is not much happens. So each dot here is a neuron, and these control neurons stay distributed around that unity line. But the deprived neurons, their activity drops below that unity line. The whole distribution seems to shift down. Okay, so now we can do the same thing, only we can do it for this later time point. So now we're going to plot baseline for each neuron against its activity during late monocular deprivation. And this is what we saw. So the deprived neurons are the purple guys, and you can see they've moved back up along that unity line. And in terms of variance, it looks pretty similar to what we see in the control distribution. And that's just one way of quantifying that. So really, neurons seem to come back quite close to their own baseline firing rate. So what this really suggests is that at least in this direction, neurons really do behave as though they have an individual set point. But now this raises the question of whether this process is bidirectional. A good homeostatic mechanism should be able to regulate things in both directions. And a graduate student in the lab, Alejandro Torrado Pacheco, spent a fair amount of time messing around with paradigms to try to enhance activity in V1. That turns out to be tough to do, but eventually he settled on a really simple paradigm. And what he did was exactly what Keith had done. He closed an eye, waited enough time for that homeostatic regulation to happen. And then at this point, he just reopened the eye. And his reasoning was that, at the end of this period of monocular deprivation, presumably there's been homeostatic mechanisms that have kicked in, that have enhanced the intrinsic excitability of the local network. So if we now restore correlated drive to that network, perhaps we could drive, some sort of LTP-like process that would potentiate firing. And then we could watch whether it would come back in the other direction. So now he's doing the same kind of experiment, only he is recording for 12 days. And this is a very difficult experiment, but he was able to get enough cells that we could see the same pattern we'd seen before, which is a drop and a restoration. And indeed, when he reopened the eye, he saw this potentiation, and it seemed like things were coming back. So now what he did was just focus in on this period of time, and so that we could follow a larger population of neurons and then ask what happens. So this is actually showing you the ensemble mean. So it's each neuron's normalized to its own baseline. And in the control hemisphere, when you reopen the eye after monocular deprivation, you see not much happens at all. But in the deprived and reopened hemisphere, you see this potentiation of firing that then comes back at the population level. And now if you look at the behavior of individual neurons, this is a different way of looking at it. So the lines are connecting the same neuron here. You can see, most of the neurons show this potentiation, not every neuron. Some kind of go rogue, but most of them show a potentiation and also then show a return back towards baseline, and that quantifies how close they get. And it's basically about as close as you see in the control distribution. And really, if you model this, you can only really get this if neurons are aiming back towards their original set point. Okay, so what I've told you so far is that pyramidal neurons in V1 seem to regulate their firing around an individual firing rate set point. And I'll just mention that firing rates aren't the only thing in these networks that are under homeostatic control. So the correlation structure of the local networks is also homeostatically restored. That's some work we did in collaboration with Juliana Georgieva. And also the closeness of these networks to criticality is also under homeostatic control. And that's something that Keith Hengen kind of spearheaded in collaboration in his new lab. And so this raises just all sorts of interesting questions that we're busy working on. One of them is, how are these sort of individual set points built? And we don't have any kind of complete answer to that yet. But Nick Trojanowski in the lab has been working on a method for being able to ask these questions that is working pretty nicely. We just published a paper on that. And then the other sort of interesting thing that this raises is the question of whether, if these neurons have these set points and these set points are important in some way for network function, then are there disorders where you can show that these set points are messed up? And let me see, what time is it? I think maybe I will just zip through this part to get to something more fun and just tell you that
Can I ask another question though?
Yeah, please ask questions.
Yeah, so your data absolutely shows that that these neurons have individual set points, and I'm actually amazed that their set points vary so over several orders of magnitude in terms of frequency. But I also noticed that they also seem to differ in how quickly their activity decreases and then recovers. And did you analyze that, and did you see any relationships? Is there any relationship between the speed of adjustments in a given neuron and its set point?
Yeah, there doesn't seem to be. So in terms of the drop in activity, that's pretty interesting. And we don't fully understand the mechanism that's driving the drop. But one of the things you see is that there's a population of neurons that drop So if you close an eye and you wait until the next light period, when the lights come on, you see a population of neurons that drop at that point during the first light period. So it's driven by probably decorrelated visual input through the closed eye. Right? But there's a population of neurons that don't drop until the second light period. And they seem to be intermixed. We don't have enough cells that we can parse them very many times and still have, any kind of statistical power. So it's hard to ask. There's other ways of getting at that now, maybe, that would let us start to ask those questions. So we don't quite understand what it is that makes some drop more quickly than others. But presumably it's something about the local connectivity and what's happening to the local network. And in terms of the recovery, that is much more, similar across neurons. I mean, it really does seem to be slow. And as near as we can tell, it seems to, the drop happens pretty quickly. And, you could imagine something is going on like, LTD saturates or something. So at that point, you start to see the recovery happening. So it seems like it just unfolds slowly from whenever that, that maximum drop happens.
Hey, Gina, since you've been interrupted, I don't quite know how to ask this question, but you'll understand it when I ask it, no matter how I ask it. And that is that you have a sort of a confound in terms of understanding mechanism, because you have an individual neuron set point, that's fine, but there's also, it's in the context of all the circuit activity. And while the neurons return to their original firing rate, how they get there in terms of the dynamics and in terms of how much of it is the set point of the neuron versus the set point of the circuit, since you probably already know that different neurons you're recording are parts of different little circuits. How can you tease out the component that's due to the set point of the neuron versus the set point of the circuit?
Yeah, I mean, I think it's a great question. And, I don't think we can at the moment. I mean, I think right now what we can say, some data I just whizzed past and I'm not going to show you, just because I'm going to talk about sleep and wake instead, because maybe it's more fun. I don't know. Maybe not. We know that manipulations that block synaptic scaling and intrinsic homeostatic plasticity, which are two mechanisms that together seem are correlated with that increase in firing, the restoration of firing during upward firing rate homeostasis. Manipulations that block the recovery of the network. So that argues that at least a major part of it is, a measurable part of it is actually these cell intrinsic mechanisms that are bringing neurons back to an individual set point. But I don't think it doesn't mean it's all of it. And for instance, the role of inhibition is very poorly defined at this point, in part because it's really complex. There's lots of cell types. The inhibitory networks themselves are very complicated. They inhibit each other. And so disentangling all of that at the network level is a lot of interesting work that just hasn't been done yet. So I think, your point is right on. Both things are going to be happening here. Figuring out all of their relative contributions will be difficult. But we can show there is an impact of blocking synaptic scaling in, I mean, of course, we're blocking it very, very broadly also. So we're affecting network compensation, not just individual compensation. So that's the best I can tell you at the moment. But I think that's the way these mechanisms work. They are embedded in these complex networks, and lots of changes are happening at lots of places in the network. So
Thanks.
Gina, I have a question. Do you have any insights about what happens to not just normal synaptic interaction, but things like synchronization for oscillatory states and things like that?
We haven't actually, we could look, maybe we could. We haven't looked much at, we do have local field potential recordings that we could potentially get some of that information from. It's a little tricky to compare them across these long periods of time. So, so we haven't tried to do that. One thing we have done is look at sort of pairwise correlations of neurons in the network, which is one sort of very simple way of starting to ask about network structure. And there, what we can see, this was with Juliana, we can see that when firing rates drop during early monocular deprivation, we see a reduction in those correlations. When they come back, the correlations go up again, and the correlations seem to be preserved from one, from control to these late MD time points. So in other words, these homeostatic mechanisms seem to preserve those network correlations. They don't destroy them. But that's about as far as we've gone in that direction.
Let's do a little sleeping.
Okay, let's go on to, let's see, what did I put in next here? Ah, yes, behavioral state. So one of the wonderful things about these recordings is that these animals are just freely behaving. They're doing their own thing. They're sleeping, eating, waking, running around. And we have video recording of all of this, as well as the spiking activity in V1 and local field potentials and all of that. So we can very rigorously characterize things like their sleep and wake states. And of course, we know when in the circadian cycle we are and all of these other things. And so we got really interested in the question of whether sleep and wake might in some way gate the expression of homeostatic plasticity. Now, why might we think this? I mean, in some ways, you would predict from our culture work where, presumably there isn't much in the way of behavioral state or neuromodulation, that the homeostatic mechanisms are expressed just fine. But one reason we got interested in it is this, this idea that's been around and very influential in neuroscience for what sleep is good for. So one idea I think all of you have heard is that, sleep is for memory replay and memory consolidation. And by this measure, sort of Hebbian mechanisms are mostly driven by sleep. But there's this other idea that's been around for a while and very influential in the sleep field. And this is the idea that sleep is really for homeostatic resetting of excitability. And this is an idea that Giulio Tononi and Chiara Sorelli have kind of championed, the sleep homeostasis hypothesis. And the idea, just put really simply, is that when animals are awake, they're running around, they're encountering things in the environment. This drives a lot of LTP and learning. And what that tends to do is, saturate synapses. And so what sleep is for homeostatically renormalizing excitability. So we sort of realized, and this idea is really that, and there's some data supporting this, is that wake just drives long, sort of widespread increases in excitability. And that when animals fall asleep, simply the process of being asleep actually drives synaptic downscaling, as they call it. Okay, so we realized we were in a nice position to test this because we had all of this beautiful behavioral data, and we actually set the experiments up so that we would be able to categorize sleep carefully and test this. So again, something you probably don't know, I think everyone has heard that rats and mice are nocturnal, and that idea is just wrong. So kind of throw it out. This just sort of shows you sleep and wake behavior of a typical Long Evans rat. Basically, during the light phase, their sleep bouts are longer. There's less time awake, but they cycle between sleep and wake during both parts of the circadian cycle. So we thought one possibility is we would see this firing rate homeostasis happen as just a continuous function of time, that wouldn't be modulated by sleep and wake. But according to the sort of shy hypothesis, well, the idea then is that homeostatic compensation would only happen when the animals are asleep. And when they're awake, maybe you would see no change at all in firing. So Keith did, and Alejandro also, just a whole bunch of different analyses to try to get at this. This is one analysis that we did. So this shows you one of these neurons where during monocular deprivation, they drop and then the firing rate rebounds. And if you sort of blow up this period of time, we can determine when the animals are in these different states, REM, non-REM, active or quiet wake. So what active wake means is the animal's actually moving around, sort of engaged in its environment. Quiet, it's just sort of sitting quietly, spacing out. So we can look at this during this period of rebound, and we can divide, we can find, for instance, all the periods of active wake, and then pull those out. And then we can, because each of them is a different length of time, you have to do something complicated, which is sort of normalize them on the time axis. So we express that as time in epoch. And once you've done all of that, you can then average them and just ask whether we can detect any kind of change across that type of state. And so when we did this in the control hemisphere, now remember, we have electrodes in both hemispheres. So the animals are running around and both hemispheres are sleeping and waking together, and the animal and are experiencing all of the same behavioral state changes. So this makes for a really nice control. And what you see in the control hemisphere is not much at all. So there's no change in firing across the epoch as we would expect. But if you look in the deprived hemisphere, as firing rates are coming back, what you see was really pretty striking and surprising to us. So in the sleep states, that's the blue and the purple here, we saw no change at all. And it was actually only in active wake that you could see this really robust increase in firing. So it seems that upward firing rate homeostasis is completely absent during sleep and is turned on during primarily active wake. We see a little of it in quiet. And then you can do fun experiments like you can try, well, we don't call this sleep deprivation, we call this wake extension experiments. So in these experiments, what we did was to take every natural, every time the animal would wake up, we would just keep them awake for a full hour. Then we would let them fall asleep, sleep naturally, wake for a little bit, go back to sleep. And then the next time they would wake up, we would keep them awake for an hour again. And we would do this just by giving them a new toy or tickling them a little bit with a paintbrush, various things that the animals find non-stressful and kind of fun. So, so we can keep them awake without cumulative sleep deprivation, which is an incredibly stressful manipulation. And then we can look at when we can see this increase in firing. And what you see is in the deprived hemisphere, we see it during these periods of extended wake very robustly. We don't see anything in the control hemisphere during those same periods of extended wake. And then if you look during these very short natural wake episodes, we don't see much, and that's because they're so short. And you really need at least 5 minutes in a natural waking state before you start to induce any homeostatic compensation. So again, this really suggests it's happening during these periods of wake. Okay, well, what about downward firing rate homeostasis? Oh, and by the way, I just want to say one thing that we found very, very robustly, both in visual cortex and also in prefrontal cortex, is that sleep by itself, sleep and wake by themselves do not drive synaptic changes. We only see changes in firing rates or in synapses, another study I'm not showing you here, when we have perturbed the system to induce homeostatic plasticity. So sleep by itself doesn't downscale synapses.
Gina?
Yeah.
So in the previous slide, does that mean that the recovery to the previous level of activity is faster when you give the extended wake? In other words, if you compare to animals.
Yes, although we don't have a ton of animals, so I think this was it 4 or 6 animals we put through the sleep deprivation. I mean, they're very difficult experiments to do. So at the level of trying to compare the 4 to the statistical power is not great. So one would want a higher throughput way of trying to ask that, but the data are consistent with that. Okay, so what about the other direction? So Alejandro did kind of the same thing for this downward firing rate homeostasis. Actually, this is a slightly different analysis that we also did earlier, but in this, he basically just scanned through the data to find every period when there was a prolonged episode of sleep or a prolonged episode of wake. And then again, could ask, when do we see a robust, in this case, reduction in firing? And what he found is that it's during periods of sleep, actually, that you see these robust changes in firing. Oh, and again, sleep deprivation really greatly reduces now that recovery. So I'll just tell you, well, actually, yeah, I guess we're kind of running low on time. The reduction in firing is sort of linearly related to the amount of time the animals spend asleep. So that's shown here. And the other thing I'll say is that it really happens across both non-REM episodes and REM episodes. So here we just analyzed firing rate before and after a period of non-REM or before and after a period of REM. And we see a drop across both kinds of sleep. So that's also kind of interesting because REM and non-REM are really different in terms of cortical activity, but they both seem to drive downward homeostasis pretty equivalently. Okay, so basically what we found is that firing rate homeostasis is profoundly influenced by the behavioral state of the animal, and in kind of unexpectedly complicated way, so that one direction of homeostasis is dependent upon active wake, and the other direction is dependent upon sleep. So this sort of raises a really interesting set of questions, I think, that we don't, of course, have the answers to yet. One is, what is the mechanism? And a kind of obvious culprit here might be the neuromodulatory state of cortex in these different states. So maybe finally we're going to get back to studying neuromodulation, what I thought I'd be studying so many years ago in Eve's lab. And we have some very preliminary data from Juliette Bodorf suggesting that cholinergic modulation might be really important for upward homeostasis. And then the other question I think is really interesting: why would these two directions of compensation actually be segregated into different behavioral states? And it seems that evolution has sort of used these behavioral states as a way perhaps of segregating different processes so they're not happening simultaneously, to temporally segregate them. And exactly why that would be, isn't completely clear. One idea we have is that maybe segregating them really allows sort of maximal direction of compensation in the behavioral state for which that becomes most important. But this is really basically kind of hand wavy at the moment. Okay. I'm gonna just to show
Can I
you.
ask one more question?
Yeah.
So didn't you show a few years ago that the upward and downward synaptic scaling are different mechanisms molecularly?
There is both, there are both shared pathways and distinct pathways. And the problem is we don't really understand the full signaling, cell biological mechanisms that drive either of them. So it's not entirely clear at the moment the degree to which the mechanisms are segregated. But that's certainly one possibility you can imagine, is that this is a way, this temporal separation is a way of preventing molecular interference or something, either between the homeostatic mechanisms or the homeostatic mechanism and something else that happens in the other state, or something like that. So I'm just going to show you two very quick unpublished preliminary things that are kind of a fun direction that we're going in recently. And we've gotten pretty far on these sort of sledgehammer visual deprivation or eye reopening sorts of experiments. It's a wonderful, venerable paradigm. We understand a lot about what's happening in V1. So it's been really useful, but also it's we've been doing it for a while, it's a little boring, but also it's, we're kind of messing up cortex. And so we wondered if there was a way of actually enhancing visual function in these animals instead. So we got really excited a while ago when Jennifer Hoy and Chris Neal's lab published this really fun paper in Current Biology showing that, and this has actually been known for a while, but she did a nice characterization of it, that rodents are not just prey, but they are also predators. And it turns out that they will hunt crickets, insects in general, but we and many people just use pet store crickets because it's easy. So we have been playing around with this, and it's looking really pretty fun and interesting. And this is some unpublished data from Brian Lane, who you may remember was a graduate student in David Schultz's lab. Okay, so just a few preliminary things I'm going to show you. This is a rat with electrodes in V1 chasing crickets. We have the setup's a little different now. We basically have an arena with 4 Arduino controlled cricket dispensers that randomly dispense a cricket from one of these places at intervals that are not predictable. And then after a while, the animals figure out that they are really interested in the crickets, and they chase them and they eat them. They capture them and eat them. And they're very good at ignoring these poop distractors. They learn how to do this. So this is an intrinsically motivating task. I mean, it's not a task, right? They want to capture the crickets and they like to eat them. So they're really motivated to do this, but they're really bad at it to begin with. So this shows you just the trajectory of the cricket and the rat sort of early on, and the rat is really running after the cricket all over the arena before it can finally grab it, and it takes them a long time initially. But after a couple of hunting sessions, they become really very good. In fact, the rats get so good that as soon as they get the auditory cue from the Arduino, from the dispenser, they start running around with their mouths open, basically, trying to catch the crickets before they even get into the arena. And this shows you proximity. So, initially they get close, but they just keep fumbling the cricket. They don't have the right, hand mouth coordination to grab it. But after they get good at it, they're very, very quick. This is, again, Jennifer Hoy showed in mice that this is vision dependent. And we are finding the same thing in rats. So if we turn off visual cortex using, dread mediated, a couple of different ways, they are much, much worse at this. So they kind of use auditory cues, but they're not great. And so we're starting to see some pretty interesting things. This actually shows you the activity of 2 different neurons over a couple of hunting sessions. And in this neuron, you can see really strong modulation of firing whenever the animal is pursuing the cricket. And this shows you across lots and lots of pursuits, there's this consistent increase in activity. And what we find is that the number of neurons in V1 that respond really consistently to pursuit goes up pretty dramatically as they get better at this task. And also the strength of that modulation also goes up. And the other interesting thing about it is that the activity of these neurons can be perturbed for long periods of time after hunting. So this shows you 3 neurons were recording continuously through 3 hunting sessions. And you can see this neuron's activity just keeps getting bumped up and stays up for many hours afterwards. Some neurons actually see their activity decrease. And so it seems like this produces some really long-term perturbations and reconfigurations in visual cortex. So one of the things we really want to know is, well, lots of stuff, but after this perturbation of firing, do firing rates actually come back to this individual set point, or does hunting kind of does it move neurons around? Do their set points change? In other words, is modulation of set point maybe one of the substrates of learning? So we don't really know the answer to that yet. This shows you, a couple of neurons. We have more now, but not enough to say yet what's going on. Followed through, baseline hunting, immediately after hunting, and 24 hours after hunting. And in some cases, the firing rates really do seem to come back very close to where they were before, but in others, they seem to be changed more persistently. So we're just not quite sure what the answer is there yet. But anyway, we're really kind of excited about this as a fun, sort of ethologically relevant paradigm for really studying enhancement of visual function and the plasticity mechanisms that get engaged. And I just like it because the rats love to hunt, and it's just a fun experiment to do. Okay, so that is, let me see. Yes, I'm going to just, if I can, go to the end. Sorry, I'm just trying to go to my last slide, which of course is always difficult to find without going through everything. There we go. Okay, so just to, I've had an amazing lab. Incredible people have contributed to this story over the years. Everyone really. I didn't get to tell you about this story from Chihang and Raul another time, but I told you a lot about the work of Keith and Alejandra, who just graduated. Again, want to mention, Sasha Nelson, really important collaborator on a lot of this work. And my other collaborators at Brandeis, Don and Steve, who we just couldn't have gone in this new direction without their help and input. And that's it. So happy to take any other.
Well, Gina, at some point, there's some pretty snide comments in the chat about sacrificing crickets as food, but which offends some of our classic invertebrate neuroscientists. And Astrid is claiming that she's going to retaliate by feeding mice or lobsters. But I would like to be serious for one more second, although I think you should take Dirk and Astrid's suggestions very seriously. And I would say that the most dramatic thing that I see in the most recent data is what the hell is happening in the motor system. So, you've got this massive change in motor performance. And so the question is, have you thought about recording in motor cortex or in basal ganglia or some other, but particularly motor cortex, because God knows what's happening on the motor side as they become useless and then become expert.
Yeah, no, I think it's great question. Obvious, we might get there. I mean, I think it'd be really fun to record from both simultaneously.
Exactly. I mean, you really know how
what's interesting, Eve, is that we don't see, like, if the animals are just running, if they just run from one side of the thing to the other, when they're not hunting crickets, we don't see these modulations. They're really specific to this motivated behavior. So it really has something to do, it's not just the motor it's not something
Well,
as
they're
simple
learning a
as-
skill. I mean, this is clearly the acquisition of a motor skill. And
Right.
it requires sensory information to acquire this motor skill.
Right.
But I mean, I think that's what makes it so, so fascinating. It'd be really interesting to see what's going on both sides of that.
Yeah, absolutely.
Whether they're matched in time or not, or, you know, I mean, there's got to be something.
Maybe we'll
What?
get there before I retire. We'll see. The other thing I forgot to show you on that slide that I think is really interesting, maybe you guys noticed it, but there's a neuron in there that responded not when the animal was chasing the crickets, but when the animal was eating the crickets. And so that sort of raises the interesting possibility that there's some kind of reward signal in V1.
Dirk does not approve of that cell. Dirk and Angsker both complained about these experiments.
Well, what can I say? They're crickets. You know.
I mean, if you use cockroaches,
You
I
might dissect crustaceans. I mean, you
I
can't get a hard
think
time
you-
about feeding crickets to my mice.
I think if you use cockroaches, you would be serving a real function.
Well, you know, crickets can jump. So it sort of adds a whole nother.
I know,
level.
but cockroaches are really awful. So
And if they get
feeding
cockroaches
cockroaches,
if they get loose in the lab, that would be very bad.
that's
Actually, we already have some crickets loose in the lab, which is not good.
Actually, I shouldn't talk. I ate a cricket once.
chocolate covered or not.
Yeah, in candy, like as a lollipop, basically. It was about as pleasant as it sounds, actually. Gina, maybe I missed this, but the intermittent sleep and wake cycles are still running on top of a circadian regulation, which has its own hormonal fluctuations and things like that. So the difference is that you've shown where they're all sort of like from separated by day sleep and wake versus night sleep and wake, or
Yeah, we looked at that in a couple of ways. One was just to look at sleep and wake during either the light or the dark cycle, just pull them apart, and we see something very equivalent. And then we also just looked at, you know, the other thing I can say is that these episodes that we pull out of the long periods of wake or sleep are really distributed throughout the circadian cycle. So both of those things really suggest that there isn't a huge circadian component to this process. We really haven't seen a great, I mean, we've looked because we're interested in it, but we haven't seen much evidence of circadian modulation of the processes. So if it's there, it's pretty subtle.
Jorge will be unhappy to hear that.
No, actually I'm not. You know, Gina, Alfredo Kirkus' work on synaptic plasticity and especially lately inhibitory, and everything seems to be sleep dependent and not, I don't know if everything, but sleep seems to have a very important role to play.
Yeah, I mean, I would say just to editorialize for a second, I mean, there are, there's obviously a potential huge circadian sleep confound. And then a lot of times when people, oh my cat, when people are, sorry, I'm being distracted. Often people are not very careful about disentangling them. And then the other issue is often they use circadian time as a proxy for sleep. You know, so in other words, this goes back to this, are rats and mice nocturnal? You know, often the first 6 hours of the light period is considered to be sleep, and the first 6 hours of the dark period is considered to be wake. But when we do a really careful analysis, what we find is individual animals vary dramatically in when they sleep throughout the light of the dark cycle. And so you really can't predict based on circadian time whether they've been asleep or not. So yeah, so the field is a little bit of a mess when it comes to disentangling those things. And then
Gina, are you saying that a lot of the circadian results from rodents actually mean that they are sort of an underestimate because of that 60, 40, 40, 60? So anything that is really just regulated by sleep-wake cycle but is presented as a circadian regulation is a complete underestimate then, right?
I think
Because the differences are a lot smaller
I think in a
over
rodent,
circadian time.
unless you're carefully measuring both, circadian time and the sleep wake states in your individual animals, you can't separate the two things, because same animal, you'll find one day it's like sleeping nicely for 4 hours when the lights come on. The next day it's running around like a maniac for and why, I don't know. That's just the way they are. They don't have consolidated sleep the way we do. And so anyway, so I think a lot of work in the field is really confounded. So things that are claimed to be sleep might be circadian and vice versa, because they haven't been disentangled very carefully.
Just a comment. I think what you're doing of recording in vivo is super important in this sense, right? Because every slice work, and I've been working with Alfredo a little bit on slices, will only reveal things that appear to be circadian, right? Because we have no clue when the slice doesn't sleep. So how do you know whether you're catching the slice at the moment where that would have been sleep, right, or any other state?
I mean, you can. So one of my students, Brian Carey, has done experiments where he's monitored the sleep of the animals very carefully, and then he can do this in real time, so he can watch as they accumulate sleep or wake, and then sacrifice them at a time point when they've had a sleep dense experience or a wake dense experience, and then do the kind of slice experiments. And so so that is possible. It just requires a lot more work. You have to stick your EMG electrode and your LFP electrode in there and monitor the animals for a couple days, and carefully do the sleep wake analysis.
And do things match what you expect from in vivo work? How do you evaluate that, though?
Yeah, pretty well. I would say so Brian actually did this too. What Brian was trying to ask was whether this idea of the sleep homeostasis hypothesis, that just being asleep is enough to downscale synapses and change excitability, was right or not, because our firing rate data was really arguing against it. So he did what I just told you. And then the other thing he did was he injected a virus with channel rhodopsin into the LGN, waited until those terminals expressed channel rhodopsin up in V1. Then he implants an optrode into V1, and he can activate those synaptic terminals and look at local field potentials in V1. And so, it's not a perfect measure, but it's a way of trying to look at synaptic strengths in vivo, which is kind of hard to do. And both of them show the same thing and is consistent with our firing rate data, which is that sleep and wake don't seem to drive constitutive changes in synaptic strength. We see some very subtle circadian modulation that's pretty similar maybe to some of the things that Alfredo has seen. But we don't see much evidence of sleep driving changes by itself. So it seems to gate the plasticity. It doesn't, it's not instructive for the plasticity, if that makes sense.
We could probably keep Gina for another 3 hours, but I think we probably have to release her, because we have to have that. I was told we have to have our little PI meeting now. So Gina, I'd like to thank you so incredibly much for a really beautiful talk. And any of you who have further questions can obviously just send them to Gina or whatever. But I think nothing could have been a nicer way of spending the gray day. And so